Alaikum and welcome back to another video. In this video, we are going to discuss about the process of cell division, and we are going to discuss a process called mitosis. Earlier, we had a brief introduction of cell division. So today, let's talk about mitosis. Now, when we speak about mitosis, we often refer to it as an equational division. So it means that during the process of mitosis, as the cell divides, it maintains the parental chromosomal number. So if you have a diploid cell and it's undergoing mitosis, the chromosomal number it will remain unchanged. Okay. So in the previous video, we have discussed the different phases and we saw that the broad categorization of the cell cycle phases into the interphase and the M phase. So interphase, it has G1 phase, S phase and G2 phase, broadly saying that this is the phase in which the cell is preparing for the division. And now you can see that in this interphase, in what condition does the genetic material appear? So you can see that it's present in a highly decondensed state. You will see that the intercalations, even after the chromosome, even after they are replicated in the S phase, you will still find they are present in this same decondensed state. Okay. Now, so this was about the interphase. You can see that the nuclear membrane that is intact and the genetic material is present in a very decondensed state. Now, after the interphase, the cell enters into the prophase. Okay, so now this is the first phase of the M phase. Okay, so in the prophase, the first and foremost thing which we can see is the disintegration of the nuclear membrane. You can see here. Okay. The second thing which happens as the cell cycle proceeds through the prophase, it begins to condense. Okay, and the protein which helps in the condensation of the genetic material, that is condensin. Now there are two proteins, one is cohesin, another is condensin. Condensin helps the genetic material to condense, and cohesin, it helps these sister chromatids to be attached to each other. And this is the same cohesin which needs to get dissolved in order for these two sister chromatids to separate and then move into their respective daughter cells. So that was about the prophase. And by the time the cell reaches the cell division, as the cell proceeds through the cell division, by the time it reaches the end of the prophase, you will see that these chromatids, they will be present in an extremely condensed form. Now, after the prophase, the cell, it enters into the another state, that is the metaphase. And in the metaphase, there is another important property or another important process which happens, that is called as the bivalent attachment. Let's understand the term bivalent attachment and monovalent attachment. So in the metaphase, you can see these microtubule organizing centers, they are irradiating these microtubules, and these microtubules, they are here attaching to the kinetochores which are present on the adjacent sides of the centromeres. So one over here and another over this side. So basically what will happen as this cohesin ring is broken down, so these sister chromatids, they will separate from one another and they will move into one to this microtubule organizing center and another to this microtubule organizing center. So this formation here, this is what we call as the bivalent attachment. Okay. Now see, in this case, in this sister chromatid, you can see that it's attached to both the microtubules from here to this center, microtubule organizing center, as well as to this microtubule organizing center. Okay, so this is a bivalent attachment, and this is very crucial in order that each cell receives the genetic material. Okay, but then there is the monovalent attachment. Now, what if in this condition, both the sister chromatids, they are attached to a single microtubule organizing center. And what will happen? In this case, one of the cells will receive the entire sister chromatid, and one of the cells, it will be devoid of it. So that is the monovalent attachment. Obviously, that will be a deleterious condition for a cell. So we want this bivalent attachment to happen in order for the cell division to proceed normally. Okay. So this is what happens in the metaphase. In metaphase, you have the proper alignment and the microtubules attached to the kinetochores of the sister chromatids. Now, after metaphase is completed, we have another phase, that is the anaphase. Now, during anaphase, the chromosomal segregation that is triggered by the proteolytic destruction of the protein, we have earlier also seen the quiescent. So the same cohesin rings which were holding these sister chromatids together, these are now broken down. And they will facilitate the movement of these sister chromatids apart from each other and then finally getting into the each daughter cell. So this completes the process of the anaphase. And now you can see the movement of these towards the opposite folds. One moves to this direction, another it moves to this direction. So they will move in each directions. Now comes another state, that is the telophase. Now this is the final step of mitosis, during which you can again see the same phenomena. So this is a kind of similar state here. Now what you can see is that the genetic material is distributed. You can see the microtubule organizing centers are there. Genetic material is distributed. And now what happens is the nuclear membrane, it begins to form around each set of the segregated daughter chromosomes. Okay. And now this process, it is followed by the cytokinesis. So whereby here you can see that the nuclear membrane is formed in a proper manner and the cell, and now it proceeds for the division of the cell as well. Okay, so first the formation of the entire nucleus happens and then the cytokinesis takes place. So that was about the process of mitosis. And in the next video, we are going to discuss about the process of meiosis.